Welcome to the PubMed Daily Review for March 03. Today we scanned 2,669 new open access papers. Here are the top 3 selected for detailed analysis. Up next, our first paper out of 3, Functional Correction and Genome Integrity with Duplex Base Editing of Beta-Thalassemic Hematopoietic Stem Cells by Nicoletta Y., Papayawanu and Karsten W. Letterer from Molecular Genetics Thalassemia, the Cyprus Institute of Neurology and Genetics, Nicosia, Cyprus. Let's break down its key contributions. Beta-thalassemia is a major global health challenge, affecting millions. While gene therapy offers hope, concerns remain about safety and efficiency. This study explores a new generation of gene editing tools, base editors, which aim to correct the genetic defect without causing double-strand breaks, potentially leading to a safer and more effective treatment. To tackle beta-thalassemia, researchers used patient-derived stem cells. They employed a technique called duplex base editing, which targets 2 key genetic sites simultaneously. This was directly compared against single base editing and the current standard, CRISPR/Cas9, which uses double-strand breaks. The goal was to see if this new method could effectively boost fetal hemoglobin and, crucially, if it was safer. The results were quite promising. Duplex base editing led to a significant 6.4-fold increase in fetal hemoglobin, which is key to correcting the anemia in beta-thalassemia. This was achieved with a very low rate of unwanted DNA insertions or deletions, less than 1%. Importantly, this also led to a significant reduction in adult hemoglobin, indicating a shift towards the beneficial fetal form. So, what does this mean for patients? This study highlights duplex base editing as a highly effective and potentially safer therapeutic strategy for beta-thalassemia. By minimizing DNA damage, it could offer a more robust treatment option compared to existing gene editing methods. While these findings are exciting, further clinical trials will be essential to validate its long-term benefits and safety in humans. Up next, our second paper out of 3, CRISPR-Cas9-based gene editing as a proof-of-concept approach in an inborn error of immunity caused by a DCLRE1C variant by Tugs Duran and Ismail Risley from faculty, Department of Medical Genetics, KTO Karate University, Konya, Turkey. Let's break down its key contributions. Many children are born with inborn errors of immunity, leaving them vulnerable to infections. Current treatments, like stem cell transplants, are effective but carry significant risks. Gene therapy, particularly using CRISPR-Cas9, offers a more targeted approach to correct the underlying genetic defects. This study explores its potential for a specific type of immunodeficiency. Researchers took T cells from a patient with a specific DCLRE1C gene defect. They used CRISPR-Cas9 technology to precisely edit the faulty gene. After editing, they analyzed the DNA to confirm the correction and then tested the cell's function to see if the editing had a positive impact. The study confirmed that CRISPR-Cas9 successfully corrected the DCLRE1C gene sequence in the patient's T cells. Following the editing, there was a significant increase in CD25 expression, a marker of T cell activation, showing improved cellular function. While DCLRE1C mRNA levels didn't change, the protein function showed improvement. This research demonstrates the potential of CRISPR-Cas9 gene editing as a therapeutic strategy for specific immune deficiencies. Although the functional improvements were promising, further work is needed to achieve full restoration of T cell function. This study is a crucial first step towards developing gene-based therapies for patients with DCLRE1C variants. Up next, our third paper out of 3, SLC7A11-mediated cysteine/glutamate antiport reprograms macrophage polarization and ameliorates atherosclerosis by Shuaishuai Zhou and Fudi Wang from Department of Nutrition and Health, China Agricultural University, Beijing, China. Let's break down its key contributions. Atherosclerotic cardiovascular diseases are a major global health concern. Macrophages within arterial plaques are key players, and their polarization state significantly impacts disease progression. While amino acid metabolism is known to influence macrophage behavior, the specific role of the cysteine/glutamate antiporter, SLC7A11, in atherosclerosis remains unclear and warrants investigation. To explore SLC7A11's role, researchers used APO knockout mice engineered for macrophage-specific SLC7A11 overexpression. These mice were fed a high-fat diet to induce atherosclerosis. They also utilized bone marrow-derived macrophages to investigate the molecular mechanisms. Key techniques included histological analysis of plaques, gene and protein expression studies, and specific assays to assess macrophage polarization. The results were striking. Mice with increased macrophage SLC7A11 showed a 61% reduction in atherosclerotic lesion area. This was accompanied by decreased expression of M1 pro-inflammatory markers and increased expression of M2 anti-inflammatory markers. Mechanistically, SLC7A11 overexpression reduced STAT1 phosphorylation, associated with M1 polarization, and enhanced STAT6 phosphorylation, linked to M2 polarization. This study reveals that SLC7A11 is a key regulator of macrophage polarization and has a significant protective effect against atherosclerosis. These findings highlight SLC7A11-mediated amino acid metabolism as a promising novel therapeutic target for cardiovascular disease. Future work should focus on developing strategies to modulate SLC7A11 activity in patients to combat atherosclerosis.